This presentation will be on Craig Mello and Andrew Fire, who won the Nobel Prize in Physiology or Medicine in 2006. Craig Mello was born in New Haven, Connecticut, and moved to Fairfax, Virginia at a young age so that his father, a paleontologist, could follow his work to the Smithsonian Museum of Natural History. Mello began his education at a private school when he was 5. He reported that he did not do well in school up until about 7th grade. He much preferred being outside exploring. He went to high school in Fairfax, where he took every science class he could and became a very serious student. From there, he followed in his parents' footsteps and attended Brown University, where he studied biochemistry and molecular biology. He then went to graduate school at the University of Colorado, Boulder, where he worked in a lab to identify elements within C. elegans that directed the replication and partitioning of chromosomes in the worm. He then followed his instructor to Harvard University to continue research. He then met Andrew Fire, who was working on similar techniques, and collaborated, eventually making DNA transformation in worms a routine procedure. After Harvard, Mello moved to Seattle, Washington to work under direction of Jim Priest at the Fred Hutchinson's Cancer Research Center, where he helped identify genes that worked as regulators of early development in C. elegans. Andrew Fire, on the other hand, was born in Santa Clara, California in 1959. He attended K-12 in Sunnyvale area schools and received his A.B. degree in mathematics from UC Berkeley before earning a Ph.D. from MIT. He then received training from a C. elegans group at the MCR Laboratory of Molecular Biology in Cambridge, England. While there, he initiated research into the improvement of technology for the expression of foreign DNA in C. elegans. After work in Cambridge, Fire moved to Baltimore, Maryland to work at the Carnegie Institution of Washington's Department of Embryology as a staff associate, which meant that he could hold an independent research position where he could develop research programs without having to worry about academic obligations. His group focused mostly on developing new DNA transformation technology and conducted studies on the molecular basis of gene activation in muscle cells. As previously stated, Fire and Mello received the Nobel Prize in Physiology or Medicine in 2006 for their discovery of RNA interference, gene silencing by double-stranded DNA. RNAi plays an important role in both gene regulation and cellular defense of viruses. A number of plants and animals have developed naturally occurring RNAi that acts as an antiviral that encodes short segments of RNA that are complementary to viral sequences, in turn deactivating them. Synthetic double-stranded RNA can be used by researchers to manipulate gene expression, which gives them clues to understanding gene function. Craig Mello put it this way: with RNAi and the completion of the genome sequences for humans and numerous other organisms, we now have unprecedented opportunities to develop new life-saving therapies and to advance the basic understanding of our biology. We humans have a potentially very bright future. The biological mechanisms at work inside our cells are truly ancient and remarkably stable, more stable even than the positions of the continents and oceans on the face of the Earth. In describing the science behind RNAi, we must first define a few key terms. The first is RNAi, which stands for RNA interference, a genetic regulatory system that functions to silence the activity of specific genes. dsRNA stands for double-stranded RNA. siRNA is small interfering RNA, which is derived from longer synthetic double-stranded RNAs, which are usually approximately 21 nucleotides long. MicroRNA is derived from the naturally occurring pre-microRNA, which is transcribed in the nucleus, folded, and processed as double-stranded precursor microRNAs, which are also approximately 21 nucleotides long. Dicer is an endoribonuclease protein that cuts RNA into short segments, and RISC is an RNA-induced silencing complex, which is a combination of RNA, guide strands, and proteins. Before Fire and Mello's work, double-stranded RNA had been found to be responsible for inducing viral resistance by acting as a trigger in animals and plants. This shows that it is a natural system that has been acting in cells for a very long time. Cells respond to double-stranded RNA by producing a signal that would tell the organism to be resistant to viruses. The top line in the diagram to the left shows a highly virulent virus that is lethal to the animal. The second line shows a much less harmful virus, which causes little harm to the animal. The third line depicts a scenario in which the animal is first exposed to the less virulent strain, which then leads to an immune response that protects the survival of the animal when later presented with the more deadly virus. Early on, Fire and Mello micro-injected DNA, RNA, proteins, or virtually anything into worms using a glass microneedle. They then studied the response this injected material had on foreign DNA or RNA. One of their first experiments aimed to turn up or turn down gene expression for specific genes. In the experiment, they took a gene called UNC22, which in absence caused twitching in the worm. They injected a DNA promoter which allowed the RNA synthesis of UNC22 to begin, which made the antisense transcripts of the gene, to in turn shut down the gene. These transgenic animals which had the injected DNA showed decreased activity of the gene in question. They hypothesized that the antisense DNAs they injected were finding their antisense equivalents by base pairing to the gene and decreasing its activity. Although they could see the effect that this injected DNA had, they did not understand how the interference was working. Through further study, they discovered an enzyme complex called dicer, which was responsible for cleaving the double-stranded RNA into the smaller si or microRNA fragments, which were in turn part of a second enzyme complex called RISC. RISC is used as a guide that travels around the cell looking for target RNA. When the target RNAs are found, they are cleaved by RISC enzyme activity, which leads to the degradation of the target RNA. This diagram on the left shows two pathways for RNAi. RNA can occur either naturally or synthetically. Naturally, you have pre-microRNA, which is then turned into microRNA via dicer, or synthetically, you start with double-stranded RNA and get turned into small interfering RNA. Both of these smaller RNAs are roughly 21 nucleotides long, and after they are cleaved, RISC combines to the cleaved RNA strands with a protein complex. This complex then complementary base pairs to the target mRNA and then cleaves it so that it can be further degraded. The target mRNA is thus non-functional or silenced. As we have learned, there are many ways that eukaryotic cells control gene expression, and RNAi, which uses small RNA molecules to direct gene silencing, is one of them. Although Fire and Mello discovered RNAi and won a Nobel Prize for it, it is important to note they had a lot of help and guidance from various scientists in the scientific community throughout their careers. A quote by Andrew Fire regarding the possibilities of RNAi in a clinical setting is as follows. He asks, "Will direct administration of interfering RNA be a useful clinical tool? If a person has a virus infection, why not use double-stranded RNA corresponding to that viral sequence as a drug to treat the person? If a person has a tumor, why not take the gene that's essential for that tumor and administer the double-stranded RNA corresponding to that gene to shut down the growth of the tumor?" The information shared in this presentation can be found at the sources listed. Both Dr. Mello and Dr. Fire's full Nobel lectures can also be found on nobelprize.org for further reference. Thank you for joining me.